So in today's lesson, I'm going to be moving on from glycolysis into the next stage of cellular respiration, aerobic cellular respiration, and I want to discuss pyruvate oxidation. So you'll remember that the primary product resulting from glycolysis are those two pyruvate molecules. That is what is remaining now of our original glucose molecule. And so now we want to sort of track, well, what is happening to that 3-carbon molecule once glycolysis is over. So you'll remember that glycolysis is all happening in the cytoplasm of the cell. We know that, from grade, I want to say 6 or 7 science, that the mitochondria is, the powerhouse of the cell. It's responsible for manufacturing energy for the cell. But we really haven't touched upon that yet in cellular respiration. So now I want to go ahead and start that conversation. So the mitochondrion, you'll remember, is a molecule that looks a little bit like a bean of sorts, and it's a double membrane molecule. And so it has this sort of folded inner membrane and smooth outer membrane. So we'll often refer to the different parts of the mitochondrion as, the intermembrane space, which is the space between the outer and inner membrane. And we'll also refer to the matrix, which is this fluid-filled interior portion of the mitochondria. Now, another word that may be used is cristae. Cristae just refers to the folds, the foldings on that inner membrane. Okay, so the mitochondrion. So stages 2 and 3 are going to occur in this double membrane organelle. So what do we mean when we say stages 2 and 3? Well, we're talking about pyruvate oxidation, and we're talking about the Krebs cycle. So we know that if a cell is eukaryotic, that it will have organelles and it will have a membrane-bound nucleus. But in this conversation, we're really only talking about what happens in eukaryotic cells. The conversation around prokaryotic cells will happen a little bit later. Okay, so as we know, as I mentioned before, well, the mitochondrion is the powerhouse of the cell, but what does that really mean? It means that this is where that key molecule, that key energy storage molecule, ATP, is being manufactured, and in large amounts. So though we've already discussed it, here are some points for your notes if you want to jot them down just around the structure and makeup of our mitochondrion. Here they are. One thing that I didn't mention that I think is worth mentioning is that, as with most membranes, we're going to see some proteins and some enzymes embedded in the membranes as well. But the ones in the inner membrane are of particular importance, and we're going to see how important they are when we get to the final stage of cellular respiration, where we discuss the electron transport chain and chemiosmosis. Okay, so on to our discussion around pyruvate oxidation and what is going on in that process. So maybe just pause the video for a moment and just take a look at the diagram and try to figure out what the heck is trying to be communicated in this diagram. All right, so I'm going to move ahead with it. So as we already discussed, we know that the product that remains from glucose resulting from glycolysis are our 2 pyruvate molecules. And so they're created outside in the cytoplasm of the cell, and so we haven't really gotten into the mitochondrion yet. Okay, but our pyruvate molecule does move through into the mitochondria via a transport protein. Those are the proteins that are sort of embedded within the membrane. Right, so here's our pyruvate molecule. Let's just take a look at it a little bit more closely. So you'll remember pyruvate has 3 carbons. So here we have 1, 2, 3 carbons, and so let's just remember the title of this note, which is pyruvate oxidation. So in this process, we can assume that our pyruvate is going to be oxidized. Oxidized by what? Well, in every oxidation, there has to be a reduction, right? So oxidation meaning the loss of electrons, and reduction meaning the gain of electrons. So there has to be some sort of equal loss and gain. So you'll notice here that I've identified our NAD plus molecule. Well, NAD plus is going from having a positive 1 charge to having a 0 charge. So NAD plus is being reduced. So what is oxidizing our pyruvate? NAD plus. So our pyruvate molecule, in being oxidized, has a carbon dioxide molecule removed. So that's why we see this CO2 evolving here as waste. So when NAD plus oxidizes our pyruvate molecule, a CO2 portion of the pyruvate is removed as waste. So that's why when we think about our overall process of cellular respiration, we think of CO2 and H2O as our products. Well, here is some of that CO2 evolving as waste when we oxidize our pyruvate molecule. Okay, so then, well, what remains? We've removed a CO2 per pyruvate, which means that we no longer have a 3-carbon molecule of pyruvate. We have a 2-carbon molecule that remains, and that's referred to as an acetyl group. Okay, so that acetyl group quickly, that 2-carbon group, combines with coenzyme A, okay, which is just a specific coenzyme that often has this little prefix CoA, or sorry, this acronym, I guess you'd call it, CoA. And so what we have as a result is a molecule that's referred to as acetyl coenzyme A. Okay, so let's recap here. I've written some of these details down. A multi-enzyme complex catalyzes the following changes. We know catalysts are super important. The carbonyl group is removed from pyruvate, and then we get CO2 evolving as a result. Okay, that carbonyl group becomes CO2. NAD plus is reduced by 2H plus atoms as it's oxidizing pyruvate. Okay, coenzyme A attaches to that acetyl group that remains, that 2-carbon acetyl group, and that forms what we call acetyl coenzyme A. All right, so now we're at the point where I want to talk about the overall chemical equation for pyruvate oxidation. So you'll remember that we started off with 2-pyruvate, and the diagram above really only talks about what's happening to 1-pyruvate, but we need to remember that we want to discuss everything per 1 glucose molecule. So per 1 glucose molecule, I'm going to begin pyruvate oxidation with 2-pyruvate. Okay, and those 2-pyruvate are going to become oxidized. And in so doing, I'm going to remove a carbonyl group. And so that ends up as being a carbon dioxide, which is a waste product. So because I have 2-pyruvate, I'm going to end up with 2 CO2 evolving at this point. Okay, so then also, well, what is oxidizing pyruvate? Our NAD plus. So again, if I need 1 NAD plus per pyruvate, so overall 2 NAD plus being reduced, which will produce 2 NADH molecules. And I'll have, remember, along with that, I'm pulling off some hydrogen, so those are just going to be there, and they're going to probably just remain in the matrix, just hanging out, ready to do more work, which we'll talk about later. So when pyruvate is oxidized and we remove a carbonyl group, we're left with a 2-carbon compound, which is called an acetyl group. So that acetyl group quickly grabs onto coenzyme A and becomes an acetyl coenzyme A. So because I have 2-pyruvate and I need 1 coenzyme A per pyruvate, okay, joining that acetyl group to become 2 acetyl coenzyme A's. So to recap, where is everything going here? Well, our 2 acetyl coenzyme A group molecules are really what are remaining now as we're continuing to break the bonds of glucose and releasing chemical potential energy along the way, right? So notice that there is no substrate level phosphorylation happening here. No ATP are produced during pyruvate oxidation. However, we do produce 2 electron carriers, our 2 NADH, which are going to go on to our electron transport chain in stage 4, and that's going to result via oxidative phosphorylation a lot of ATP, but we're not there yet. Okay, our 2CO2 are going to diffuse out of the mitochondria and just kind of, that when we exhale, when we're exhaling CO2, some of that CO2 is coming from the process of pyruvate oxidation. Okay, and as I mentioned, our 2 hydrogen ions are just going to remain dissolved in that matrix, ready to do more work potentially. So there it is. There's our overview of pyruvate oxidation. I'm going to encourage you today to just try to work through this and make sense of it, understand the overall process, and ready yourself for what is coming tomorrow. In tomorrow's lesson, we are going to talk about the Krebs cycle, otherwise known as the citric acid cycle.